The overall goal of this procedure is to inject microbubbles into isolated living mouse embryos for contrast-enhanced ultrasound imaging. This is accomplished by first harvesting living E16.5 or E17.5 embryos from a pregnant mouse. After preparing the targeting microbubbles, the microbubbles are injected in the living embryo via the placental vascular network. The final step is to image the microbubbles using ultrasound imaging. Ultimately, contrast-enhanced ultrasound imaging may be used to measure vascular perfusion parameters and assess targeted microbubble binding in embryos.
Generally, individuals new to this method will struggle with the embryo handling and injection portions of the protocol until they have had sufficient practice in applying the needle and forceps pressures required for a successful experiment. Demonstrating the procedure will be Brian Nixon, a co-op student from our lab.
To reconstitute the target-ready vials, begin by diluting 20 micrograms of the antibody solution into a final volume of 1 milliliter of saline. Keep the vials on ice. Next, fill a syringe with 1 milliliter of saline, removing all air bubbles. Attach a 21-gauge needle and inject the solution slowly into the microbubble vial. Slowly withdraw the plunger, removing 1 milliliter of air, then withdraw the needle. Gently agitate the vial and let it stand on ice for 5 minutes.